Okay, so hello everyone. I hope you can see me. And we the show begins. We will start out with our webinar. I'm Alexey Gavrilenko, Director of Marketing here at Enamine. So it's the first webinar of 2026. It's going to be a few. So let the show begins. I'm going to be sharing my screen and give a possibly short presentation. Let's see how it goes. I hope you can see my slides. So let's go. So a couple of rules of today's webinar. So as you can see, our webinar is recorded and you will be muted basically during the whole the whole process of the webinar. We will be downloading the recording of the webinar to our YouTube channel. So if you or your colleagues, obviously, your colleagues couldn't make it today, you can share the link and see the webinar itself. If you have any questions that arise during the webinar, you can tap them into down below in the question box. So don't hesitate. We will be answering the question in the end of the webinar. So we'll build the webinar in three pieces. So the first is going to be my quick introduction of Enamine and the real, couple of announcements. Then it's going to be our speakers to present having the stage, and then we'll have the Q&A session. And as always, please stay tuned and subscribe to our social media to not to miss what's going on and the new announcements, webinars, and cool events. So let's get started. Quick intro of Enamine and our company. That's is 35 years of our experiences ongoing. As you know, we have a global presence in Europe and the US, and with our team of more than almost 1,000 scientists having like globally, and our motto that we are scientists, researchers to researchers, science to science, because we are made of the same, as you wish, wax. So we are two in one. We are a catalog company and a contract research organization, having this niche portfolio to our clients and customers. And obviously, our journey started like a couple of years, almost 35 years from now, and building blocks are probably the most of our prominent products that we are known from, but the others are not the least. And just wanted to highlight that in 2025, we have our chemists have synthesized almost 26,000 new building blocks, and which are all available for the ordering. And as I mentioned, that we are a purely scientifically driven company, and we speak to our collaborators, partners, clients the same way. And all of our passion and our work are depicted in the research papers that are that year after the year they're going up. And in 2025, there were almost 130 of those in world-renowned journals. And you can actually browse them in the publications on the enamine.net. And I have spoiled today's show with having mentioned that it's 35 years anniversary of Enamine, which we are all very happy about. That we such massive amounts of time here on the scientific world, and to share this happiness and to share this commitment to the global science. We are having in 9 months, we're having a 4-day massive event in Riga, Latvia. It's called Enamine Drug Discovery Conference. We will be having world-renowned speakers in organic chemistry, medicinal chemistry, computational chemistry, and biology. So the registration is now open, and most of the speakers are all confirmed. So please stay tuned and to the updates, and you are very welcome to come and to share this nice event with us. So let's get back to the today's actual topic that we'll be covering. It's one of our prominent product, which is called Enamine REAL, and I hope most of you is familiar with Enamine REAL. Is acronym for real, readily accessible. It's an approach that gives us access to zillions of molecules that can be synthesized with a very high synthesis success rate. And by actually accessing in-house our Enamine in-house building blocks, and with the synthetic procedures that are been validated over the years and years of this scientific journey. So a very simple example, when we have an acylation of sulfonyl chloride with the amine with forward arylation with another amine that we are having, and for example, we have two building blocks, and we compare this knowledge, and we see when we have one of those that show low success rate, that we delete from the protocols and keep those who have very nice success rate. So basically, yes, basically what I would like to give in here. In terms of Enamine building blocks, that they have been synthesized. Enamine synthesized new building blocks, 1,200 new building blocks a month. And over the years, yeah, this number grows, and the previous year, and as I've mentioned a couple slides ago, that our in-stock collection of building blocks is more than 350,000 building blocks that are available in multi-gram amounts, with the most normal, like commonly used, frequently used chemical backgrounds, yeah, which are normally taken to the transformation. Yeah, and also in terms of Enamine REAL is going up in having it in the scientific presence, and the number of articles is steadily. We see the substantial steadily grows, and I'm sorry for the last minute presentation, but we have in 2025 we have the almost more than 40 articles that's taken in consideration the Enamine REAL, which is pretty magnificent if in terms of one of our prominent products. Yeah, so basically from in Enamine REAL Space, the version of 2025.2, we have added two new procedures and over 13,000 new reagents. In total, there's almost 200,000 reagents that might be used, and which lead us to the great chemical space of new molecules that can be actually synthesized in a very relatively short period of time. So this basically a more precise ways to have it. So I hope you have a chance. But all this information is can be found on the website, enamine.net/real. So everything is there, and we're happy to in quite some time in the Q1 or Q2 to receive the new update of Enamine REAL Space, and please stay tuned for the updates. In this schema, just to show you that the concept of Enamine Real, so everything starts with the in-stock reagents, yeah, the pre-plated libraries, if you wish, and going up, up, up. And if we're talking that this never-ending zillions of molecules that might be synthesized to fulfill your research. And last year we have since Enamine Real is synthesized, it's a parallel chemistry approach. Last year we have synthesized almost 300,000 new screening compounds, which is pretty significant numbers, because these compounds actually were delivered to where it came into the catalog and enlarged the catalog of screening compounds, and actually were delivered to our customers to fulfill their research. And we're really proud of this number, since eventually every year it basically have the substantial growth. And making the accessibility of Enamine Real compounds is pretty extensive, because we want to make it as accessible as possible, and basically all the information is given on the website, and there are multiple of options to actually access Enamine Real in full. We have another dedicated webinar that we have on our YouTube channel. So if you're interested and would like to have more details, you can check it out, or basically reach out to me or to our colleagues if you have any questions or you're interested in. So basically, this Enamine Real we are making this virtual real with Enamine Real, and we are dedicated to the world's scientific progress and drug discovery. And we hope one of our prominent product that I've just mentioned, that helps and makes your life and research easier. And as you know, we have multiple of collaborations and collaborators who use Enamine Real in their work, and this webinar is one of those we are having right now. And I'm very happy to welcome our today's speakers. Selena Prakahasante and Christoph Grogula, both from Virtual Discovery and our partners that we work from quite some time. And my introduction is a little bit too much, I think, in terms of time, but sorry for that. I just wanted to share the news from the previous year and the plans for this year. And sorry guys for that. But I'll stop sharing my screen and give the floor to Selena. Selena, please. You're very welcome.
Hello, everyone. My name is Selena Prakasante, as Alexi so kindly introduced. I'd first like to express a great deal of thanks to the entire team at Enamine for hosting this webinar with us, in addition to the support and partnership they provide to help us push drug discovery forward. So with that, I will share with my screen. Excellent. So this talk, as you may know, has been titled "The Era of Billion Molecule Drug Discovery: Efficient Exploration of the Enamine Real Space with VirtualFlow." This, of course, begs the question, what is the era of billion molecule drug discovery? And this is a question that guides our core methodology at Virtual Discovery. And before I answer this question, I want you to imagine that you are in outer space. Humor me, if you will, and close your eyes for a brief moment. You peer out and perhaps you see the Milky Way galaxy. Somewhere nested in this Milky Way is our sun and our Earth revolving around it. Our Milky Way galaxy has anywhere from 100 billion to 400 billion stars. And beyond our Milky Way are billions of other galaxies, making there something like several billions of trillions of stars in the universe. Now, the number of different possible molecules dwarfs the number of stars by many orders of magnitude. If you imagine the various stars you're seeing as you're in this galaxy now being different molecules, there are infinite possibilities. There are several many that could be cures to the very diseases that we have unsolved today. There may stand a molecule with an exact specificity, size, binding affinity, and many other molecular properties that could cure a disease that today has no treatment. And this is truly incredible. And what we have to do then as our task stands to find these molecules. So when we think of billion molecule drug discovery, what we are in essence doing is searching for stars in the galaxy. In terms of a well-known metaphor, you can think about it like finding a needle in a haystack. In order to do this, we focus on a core concept, specifically efficiently searching vast chemical spaces at scale to identify promising drug candidates. In particular, this aspect of efficiency at scale is important. Many of you are familiar with the state of drug discovery today. Only about 10% of clinical trials succeed. This process takes billions of dollars, with millions and millions of dollars spent along the multiple phases of this process. There are thousands of diseases that today have no form of treatment whatsoever, and we're operating at a much slower pace than is needed to address this need. There are only about tens of medicines that get actively approved each year. The EMA actively approves about 30 to 50 medicines with new active substances each year, and the FDA is operating in a similar ballpark. So we need to think about how we can operate at scale, and more specifically, how we can scale efficiently. And the process we think about at Virtual Discovery is focused and engineered to overcome these specific challenges, really thinking about how we can search for molecules in a way that is more accurate and faster. So today I'll be talking about how we are approaching our efforts on making drug discovery more efficient and operate at scale. I'll first give an overview of VirtualFlow, which is the core technology that we use. VirtualFlow has been led and developed largely in part by Christoph, who's on this call as well, and will be more than happy to answer any questions that you may have during the Q&A. After discussing VirtualFlow, I'll speak a little bit about how Virtual Discovery got started and what we've been doing in our approach to drug discovery across three key parts of this early stage drug discovery, specifically target discovery, hit generation, and lead optimization. So starting with VirtualFlow. VirtualFlow is a scalable open source platform used for ultra-large virtual screening. It was built to address the core problems that we face in traditional high throughput screening, those being the large demand on resources it takes, both in terms of time and then of course the cost associated with that time, the fact that we have a reduced scope of the space that we explore because we are again restrained by time and cost, and the fact that it finds certain targets completely undruggable. If we really want to seriously consider the ability to address and identify cures for more diseases, we cannot be restricted in terms of the targets that we explore. VirtualFlow is a scalable, open, and robust computational framework that makes screening billions of molecules practical by leveraging high-performance compute systems. It's an open source workflow, and it is used for structure-based virtual screening, and it operates with this really incredible linear scaling behavior on high-performance compute clusters. We're able to prepare and screen ultra-large libraries like spaces as large as the Enamine REAL Space with several billions of molecules. And we're also able to interface with various different docking engines when we use VirtualFlow. It fundamentally increases our access to scale, diversity, and reproducibility. So VirtualFlow is composed of two key modules, the first one being VFLP, VirtualFlow for ligand preparation, and the second being VFVS, the VirtualFlow virtual screening engine. So this first module is for the ligand preparation. We're able to convert raw input into ready-to-dock 3D formats. We support desalting, neutralization, preparing tautomers, and protonating at user-specific pHs, amongst other things. And we're also able to generate multiple output formats. Once a library has been prepared with the ligand preparation part of the module, we're then able to do virtual screens using the VirtualFlow technology. The virtual screens are able to operate with this really incredible linear scaling ability that I mentioned earlier. And you can run multi-stages of the screen, rescore, and repeat with consensus options. This figure is from the paper that Christoph is the lead author on. It was published in 2020, capturing the efficiency of VirtualFlow. I'll briefly go over how the two modules I described earlier fit into this framework. So this top portion of the diagram speaks to this ligand preparation aspect where you have a chemical space. We're able to use VirtualFlow to do all these various aspects of ligand preparation to create molecules that are then in a ready-to-dock format. Once we have these molecules in a ready-to-dock format, we can take these prepared compounds and conduct a first stage screen. We're able to take your target in various different forms, whether that's X-ray, NMR, cryo-EM, and screen that leveraging various different docking forms. We typically use QuickVina 2 at Virtual Discovery, but VirtualFlow supports over 1,500 different docking programs. So there are many others that you can consider and implement. Once you do that first stage screen, you are then at that point have a top percentage that you can take as your hits. You can go from this point to experimental validation. You can also engage in opportunities to do additional screen to get even more high accuracy docking. You could also additionally leverage VirtualFlow to do hit optimization, lead optimization, where you take lead compounds, maybe perhaps identified after that first stage screen, and those that have been verified experimentally, and then generate an analog library of those compounds, and then again, screen again. So VirtualFlow really provides different opportunities to refine and identify hits throughout the core part of the early stage drug discovery. So what happens when we actually run in scale? In 2022, Christoph and his team prepared a library of about 1.4 billion compounds. Many of these compounds were coming from the Enamine REAL database. This was in 2020, and at that time, it was one of the largest ready-to-dock libraries, and truly one of the largest virtual screens at that point that had been done. They screened 1 billion compounds against the KEAP1 protein using VirtualFlow, and they were able to identify very diverse binders with actually sub-micromolar affinity. And the most promising ligand that they identified was able to bind to this KEAP1 NRF2 interaction, and it had a dissociation constant at the nanomolar level, disrupting this KEAP1 NRF2 interaction. So truly, truly incredible. And this was in 2020. And again, just want to reiterate here with this slide, one of the biggest takeaways from this result was how scale drastically improves what we're able to find when we do virtual screens. I think this plot at the bottom is really quite incredible. We see that as we go an increase in order of 10 for the screening size, we have the binding affinity and the docking scores of those top compounds that we're identifying with the screen becoming increasing in the ability to bind to that target. With these screens operating on 100 million, even 1 billion, the difference in the docking scores of those top compounds is quite different. And we're able to see in when this goes to actual experimental validation that those hits produced by these larger scale virtual screens really are performing much better. They have much better binding affinity. Their dissociation constants are in the nanomolar, submicromolar level. So that was VirtualFlow. Moving now to VirtualFlow 2, also known as Adaptive Flow. This came out in 2022. And what we're doing here is really building on scale. So the VirtualFlow 2 gives us the ability to screen the Enamine REAL Space. This has been prepared into a library of 69 billion ready-to-dock molecules. And in order to deal with this increase in magnitude efficiently, because this is a key part of what we spoke about earlier, being able to scale but do so efficiently and accurately, we need to change the approach to this slightly. So the way that this has been done is by dividing this massive space into an 18-dimensional grid that has been split based off molecular properties such as molecular weight, hydrogen bond acceptor count, hydrogen bond donor count, and other molecular properties. And that's what you see here with this grid. Within each tranche, you then have several thousands of molecules that have similar properties. And then you can do what we call an adaptive target guided screen to identify those tranches which are best suited to your target of interest. And then we proceed to exhaustively dock all of the molecules in those tranches. And the results from this adaptive target guided screening when compared to standard ultra-large virtual screens are truly incredible. As I mentioned, we are pre-screening. In the pre-screen stage, we are taking a sample of representative molecules. And in this figure here, you see in pink the results of using this adaptive target guided primary screens. And then in blue, you're seeing your standard ultra large virtual screens. This are both screens in the final stage of docking are screening 100 million compounds. The difference is with this ATG screening, as I mentioned earlier, you're just pre-screening the space first to identify the tranches, and then you're going to select enough tranches such that you get to that 100 million mark. And as you see here, across the board for all these targets, with some interesting ones like KRAS, KEAP1, amongst all the others, that the ATG primary screens perform much better than your standard ultra-large virtual screening. So there's really the VirtualFlow 2.0 really allows us to screen larger libraries, but then also actually get better results for screening the exact same amount of compounds at the end of the day. So that's really, really quite incredible. And we've been able to do some incredible things with this. Christoph and his team did a screen of FSP1, and they were able to determine the first co-crystal structures for this target. They did a pre-screen of 12 million, which meant that they took just one representative molecule from each tranche, and then they did a primary screen of 10 million compounds. And they were able to find molecules with, again, quite incredible binding affinity and build upon the literature by finding this first co-crystal structure of FSP1 binded molecule. So the last thing I want to touch on about VirtualFlow before talking about virtual discovery is some of the ongoing work in terms of leveraging VirtualFlow with additional aspects of quantum chemistry. So most recently, in December 2025, Christoph and his colleagues published an approach that first had a training set be created. This training set consisted of 1 million molecules. A few hundreds were from experimentally verified KRAS ligands. We had 850,000 from the Stone selfies algorithm, and then 250,000 were generated using those screening processes that I spoke to earlier. This process of getting that KRAS target and then screening it against the Enamine REAL Space using VirtualFlow 2 in order to identify those top hits. And they took those 250,000 hits to be part of this training set. Of this training set of over 1 million molecules, they then used classical LSTM. LSTM is a type of recurrent neural network, combined with quantum circuit Born machines to identify the most promising compounds. And they had 15 really incredible compounds, with one of them registering at 1.4 micromolar binding dissociation. And these compounds were verified in a two-stage process involving SPR and also cell-based assays. So with that, I hope that overview gave you an understanding of what it is about VirtualFlow that really enables us to explore such large spaces like the Enamine REAL. And with that, I'm going to turn now to discussing virtual discovery and how we leverage these aspects of VirtualFlow in our work. So since our founding in 2020, the team has been using these different advanced computational methods along with state-of-the-art AI in drug discovery, as well as the various open source drug discovery tools to really identify promising leads for a wide range of challenging therapeutic targets. In 2020, the company was founded by Dr. Wagner and Dr. Gorgula. And then in 2022, we began providing leveraging VirtualFlow 2 to provide these ultra-large virtual screens of over 60 billion to our clients on top of the ultra-large virtual screens we had already been providing at that point. We continue to improve upon our methodology. In 2024, we're able to really show the efficiency of our methods when compared to other techniques people had been leveraging in machine learning or other applications of docking in the CASH challenge. And I'll speak to the CASH challenge extensively in a few slides upcoming. As we've continued throughout the years, we continue to partner, expand, and collaborate. We've partnered with various companies all over the world, whether that be an academic lab, an early stage biotech, veteran pharma companies. We're more than happy and excited to partner with anybody who's looking to identify hits for challenging targets. A brief introduction to some of the core members of our leadership team. Christoph is been mentioned, he's on this call as well. Dr. Wagner and Dr. Gorgula are the co-founders. I'm, as Alexia mentioned, the CEO of Virtual Discovery, and also just want to highlight very importantly our Chief Technology Officer, Dr. Soren Draga. So with that, I will be touching on target discovery, hit identification, and the lead optimization process. First, I'll briefly go over target discovery. At Virtual Discovery, we really tend to focus on identifying leads, but oftentimes when our clients come to us, that involves investigating a target more extensively. One of the things that we classically tend to do for many of our clients is leverage MD simulations early on in the process to better understand a target, perhaps identify cryptic pockets, allosteric pockets, anything that will help us better understand the dynamics of that target. And this is something we tend to do in conjunction with most of our virtual screens, but we can also provide this independently. Two other aspects that are interesting for our target discovery or target exploration capabilities are inverse screening and small scale screening. I'll first start with inverse screening. Inverse screening is, as the name implies, going backwards a little bit. So let's say you have a compound that you think is interesting, that you know works, but you don't necessarily know what targets it interacts with, or you want to explore what targets it interacts with. We're able to screen the entire human proteome to identify what targets in our proteome that could potentially interact with your compound. Now, moving on to the small scale screens, perhaps you're very, very early on in your process and you're still looking to validate or potentially even de-risk a target. We're able to do small scale screens leveraging VirtualFlow 1 or just very low levels of docking in order to be able to determine initial hits and compounds that could interact with your target in a very fast, low cost manner, again, to just be able to get more information on your target to then go forward. And building upon what I said, a classic case is a client may come to us saying they have a target with various different isoforms, and we need to determine which one to target. before doing a screen, we can do MD simulations on the different forms. Then we can go proceed and do an ultra large virtual screen or do independent smaller scale virtual screens, really depending on what your time and budget allow for. And we're able to have that flexibility there. Another example um, that I mentioned earlier is, let's say they have a compound that appears interesting and they want to identify potential targets. We can do inverse screening, depending on if they're interested in identifying many proteins, or maybe they just have one protein that they're potentially concerned about off-target effects for one compound or multiple. We can also do counter screens in that scenario as well. So now moving on to really our bread and butter hit identification. We're able to do many different types of scales of virtual screens, and we've done many types over the past years. We make use of this adaptive target guided screening to select the most relevant subset of molecules from these ultra large libraries, and then we're going to dock the subset. These numbers on the screen speak to the subset that we are docking extensively. We primarily tend to go for this 100 million. That was a number that was mentioned a lot in many of the papers that I spoke to about earlier. We find this to be a really great balance of expansiveness with speed. We recommend this for generating initial hits as well as for optimizing leads. But perhaps you want to be more expansive. Maybe you really want to get into this incredibly large universe that we talked to earlier. You can go higher. You can do 1 billion, 5 billion. And this is going to be the most expansive, most diverse. You can explore more molecules even more exhaustively. The trade-off here is going to be it's going to take a little bit more time. Obviously, it'll take more resources as well. And you can go in the other direction as well. Perhaps you want to explore 10 million molecules. We can do this as well. As you saw in some of the papers, 10 million was used and still had some incredible results. So again, because we're using this ATG pre-screen, we're still able to explore with a great deal of diversity, and we get great and very useful, interesting results from that. Giving a brief overview of our screening capabilities, we're able to really customize every aspect of the screen. An earlier figure showed the different types of receptor structures that are supported. Of these, I want to highlight AlphaFold in particular. I think some of the work with AlphaFold is absolutely so incredible, and we've seen internally that we're able to go off of structures with just AlphaFold and be able to produce some really great results. If you have X-ray structure, if you have crystal structures with incredible resolution, we're more than happy and eager to work with that as well. But on the flip side, if you don't have anything, but there are AlphaFold structures available, we're able to do that. And the more information we have at this stage, it's really just going to increase the different ways that we can look at this problem and we can address it. So having more information is always great as well. We're able to screen quite a variety of targets. We've in the past done protein-protein interactions, ion channels, different transmembrane proteins, and various different enzymes. In terms of molecules, we are working with these incredibly diverse libraries. Enamine REAL has so many different types of molecules, things that vary in molecular weight, vary in the amount of hydrogen bonds. We have so many different properties that are being represented by the space. And even within the space, we can optimize for certain regions of the space you wish to explore by filtering particular groups that you want to avoid. We're able to narrow down the molecules as much as you like, or really just search the entire space. In terms of libraries, we spoke to the real space. We tend to also dock the in-stock portion of Zinc 15 for our clients. And then really importantly, I want to highlight that we are able to screen custom libraries. We use that VFLP module in VirtualFlow to prepare the library for screening, and then we are able to screen it. And this is really great if you have a library that you've already prepared that you want to understand better, you want to screen against your target. This is something we're able to do. I spoke a little bit to some of the filtering, but we're able to filter across the various different properties. And we are able to support various docking programs for most of our clients use QuickVina too. Again, if there's other things that you want to explore based off what is specific to you, we can do that as well. So I want to talk about virtual discoveries experience in the CASH challenge. So the CASH challenge stands for the Critical Assessment of Computational Hit Finding Experiments. That's a lot of words for really describing a challenge that is similar to the CAST challenge, which is what AlphaFold came out of, but it's really for benchmarking prospective ways of using computational methods to identify hits for targets that really have not had much success in having hits be identified for the target. So in 2024, Christoph and Soren participated for virtual discovery in the challenge. And the target for that challenge was the LRRK2 target, which has been implicated and is believed to be associated with Parkinson's. And specifically, they were looking at this domain over here. It's highlighted bigger there, but this is that WDR domain. In the challenge, we first had 35 applicants or groups apply, providing their different proposals for how they wanted to approach this strategy. It was then narrowed down into 23. Here is our virtual discovery team here. At this stage, they went about their different methods to generate hits. These hits were then confirmed experimentally. And as you see here, virtual discovery did incredibly well, identifying in this first round 10 hits that were had some great binding activity at that WDR domain. There was, again, a second round where we were able to optimize these hits, and these hits were confirmed as SPR, and virtual discovery identified 3 here. As you see, virtual discovery did incredibly well compared to the other teams, and we were actually one of the top performing teams to leverage these docking-based methods. So again, this really, I think, in my mind, just reinforces how when you're taking a approach to searching these vast spaces in an intelligent manner with this ATG and leveraging what we know about physics, leveraging again these docking-based methods, we can still perform incredibly well and continuing to build upon and our innovations by combining physics-based methods on top of what we're doing with machine learning in ATG on top of the other methods we continue to work on, we are continuing to see great results. Now, going very briefly over the methodology we took in this challenge, the workflow we use to identify hits is similar to the process we tend to identify hits for many of our clients. We first leveraged MD simulations to get a better understanding of the target. Then we did a virtual screening with VirtualFlow 2. We selected the top molecules, applying standard filters to ensure drug-like properties. And then lastly, we optimized those molecules by generating, for identifying analogs in the Enamine REAL Space, and then screening those. So with that, I'll briefly talk about lead optimization and how we do this. I first want to ground our conversation and how we think about lead optimization. 90% of clinical trials fail, and this is a number that we want to improve upon. And we think that process starts early on in the stage. It doesn't start when you get your IND. It starts early in the discovery process. And in particular, we see a large amount of failure for clinical trials being associated, or a large amount of failure for clinical trials that are done across the entire world. We see a lot of that come from a lack of clinical efficacy, unmanageable toxicity, and just not having the appropriate drug-like properties. We're able to, in our lead optimization process, focus on critical elements that address some of these challenges, whether that be prioritizing compounds with a higher likelihood of on-target efficacy, predicting and minimizing off-target interactions with our secondary screens, with our counter screens, and refining leads based off key factors that can be filtered. In terms of our lead optimization, we primarily take 2 different approaches. The first is second stage screens, and the second is de novo molecular generation. I'll speak to second stage screens first. So this is optimizing any leads that you've already identified. We're going to generate custom analogs that are derived from the lead compounds using various different synthetic methodologies. Then we'll conduct a secondary screen on those custom analogs to identify the new and most promising hits. And then you'll proceed to validate those hits experimentally. And these secondary stage screens are something that really are able to build upon what you've already done and expand and explore the potential molecules that could increase the maybe already successful binding affinity you have identified. With that, I want to briefly touch on our de novo molecular design. We're at this point iterating upon a successful scaffold. So in this image over here, let's say you had a molecule that you found worked really well, but you think it could be improved. So like, let's say you like this molecule, but this green part over here could be changed. We are able to leverage generative models to design custom analogs based off a subset of compounds that you've already identified. We can specify quite specifically what aspects of the molecules that you wish to conserve, and then which aspects that you're open to have changed. And then we leverage the generative models to design these analogs, and then use VirtualFlow to screen those generated compounds in this active learning loop and able to identify these new molecules. So as a brief overview, we provide various different services. These ultra-large virtual screens and virtual screens is really our core service offering. We're able to screen a massive libraries and identify the hits for you. We're able to also use these screens from lead optimization, whether that be using generative models or other custom analog generation approaches. And then as you're in those early stages and you want to de-risk a target or better understand it, we're also able to do MD simulations, counter screens, inverse screening for that as well. And with that, I'd like to say thank you so much for listening to me talk about Christoph's incredible work with VirtualFlow, what we're doing at Virtual Discovery. It is such a pleasure to be able to speak to you about this. If you would like to talk to us very briefly and learn more, we're hosting office hours from 12 to 1 p.m. Eastern next Wednesday. If you perhaps have a colleague on the East Coast or West Coast, or willing to stay later, we'd love to hear from you. If you have other questions, you can contact me at any time at my email here. You can also email us at Virtual Discovery to schedule a quick chat or answer any specific questions. And I would like to also thank all of you for being here. I think what I tried to paint a picture of earlier with this massive universe of molecules is so exciting. And the future of drug discovery truly has so much potential. And I think as we all work together across the various different academic labs, companies, startups, we have so much to offer and provide to pushing drug discovery forward and identifying molecules that are potentially going to be life-changing. So with that, I thank you so much.
Yeah. Thank you very much, Selena. It was a very great presentation. Indeed, it's never been easier, yeah, and more straightforward to navigate this huge chemical spaces like Enamine REAL. Thank you very much, and I think we can switch to the Q&A session. And our beloved participants, please paste your questions on the Q&A box down below the screen. Actually, I have a couple of questions that we might want to answer and to give a quick overview. So the first one will be about the flexibility. Is the VirtualFlow able to handle protein flexibility, or does the massive scale of the screen mean we are forced to use rigid receptors? So I guess the Q&A session would be sent to Christoph. So
yeah. Hi everyone.
Yeah,
it's a great question with the flexibility. So flexibility can be included in our screens in different ways. And let me shortly share a figure on that. So here you can again see the an overview of the VirtualFlow platform, and flexibility can be included here in different ways. So one way is when you have an ensemble of conformations, for example, from NMR, which provides ensembles of conformations, or from MD simulations, which can be done before the screens. When we have such an ensemble of conformations of the protein, we can include it in the virtual screening via ensemble docking, where we dock in the end against all the conformations that we have, and then in the end create a consensus or a joint score regarding the dockings to identify the best possible conformation. Another option is to include to use a docking program that supports to model flexibility directly during the docking. So one option is flexible sidechain docking, which is supported by many docking programs such as AutoDock QuickVina, and so on. So here one can specify the sidechains exactly, the sidechains that should be flexible. And they are explored during the docking in the end. And the third option could be to use a docking method in the end that supports induced fit docking, which also goes into the direction of co-folding in the end, which is another option that the platform supports. So yeah, there are multiple options regarding that.
Yeah, that's great. So we have a question from the audience. Another one, is your full optimization process at any point, do you run a wet lab experiments? I guess there were, so.
Wet lab experiments, yes, they are of course very important. So typically, when we carry out our screens, we carry them out in multiple stages sometimes. The first stage is often, a rigid protein screen, and then when we include flexibility, we often include it in the second stage, where we rescreen the top 1 million or so compounds. At any stage, we can, in principle, order compounds from Enamine and other vendors to, in the end, experimentally validate them. And from our own experience, compounds from the first stage of the screen often work quite well in experimental validation. And we often recommend only to go to the second stage, which is more complex, if the first stage did not work well enough. And after experimental validation, then we know which hits are really binding, and then we can go further to optimization if we want to. I hope this answers the question.
I hope so as well. So I have another question from the audience. Appreciate your question, actually. In the generative method that modifies parts of a molecule while keeping some of the molecular structure immutable, how are the new compounds generated? Interesting question. Yes, please.
The new compounds are generated by existing generative models for, in the end, drug discovery that already exists, such as reInvent and so on. So we can plug this into our generative platform in the end. They generate the molecules, and then they will be fed into the ligand preparation module, 3D conformation, protonation states, and so on. And then the prepared molecules will be fed into the virtual screening module, and then will be in the end ranked and assessed regarding their binding affinities. And then the results again go back to the generative model. And there we have quite some flexibility in terms of the exact model that we want to use.
I hope this one helps. We have another question about like the pain of the computational chemists and such. There are around 1,000 docking programs. Do you find some better versus another, like more valid wet lab results? It's a good question. It's more like a question. It's more like a discussion.
So, could you repeat the question? So, is it which one to choose?
Do you find, what basically what the docking program to choose around all those programs if we're targeting more like for the web lab?
That's also a great question, and the answer really depends on the case. So, if there are no known ligands known for the target site or for the entire target, then we really don't know in advance usually which is the best method for this particular case, because each docking program can perform differently well for different targets and sites. But if there are some known ligands that you have already in the lab or from the literature, then we can at first test all our available methods first with the available ligands and see which are able to best reproduce the known data. And then we can choose the most suitable one in the end.
basically. One after another. We have a follow-up question to the first one, or similar to those one. Can you comment on what are tech advantages of your exploration method against an active learning protocol?
What type of protocol?
The advantages of your exploration method against an active learning protocol.
That's a good question. Selena, do you have any thoughts? I didn't fully understand the other method that we are comparing to.
Yeah, I'm going to take the question assuming that it's talking about our previous exploration methods that we described with VirtualFlow as opposed to potentially leveraging it in a single stage as opposed to maybe doing it in multiple stages, like in active learning. And I think that with what we do, you are able to do multiple stages of your screening. I think in the one of the papers in Christoph's Nature 2020 paper, it really clearly nicely shows how you can have kind of this multi-stage approach. Um, if the question is also potentially interested in what we spoke about with generative models versus the other methods that we use for identifying hits, I think that there's advantages to each. I think in order to be able to use this de novo molecular generation, you really need to have potentially a compound already that you've identified, and we're building and generating analogs based off that. And I'd say that's the difference between them. So they're slightly different use cases that we're using each.
Right, and to add to that, so the generative approach that we have, it's also an active learning approach in the end. It's a cycle that learns in each iteration as well. And one of the advantages of the the non-active learning method that we have, VirtualFlow 2, for example, is that we are really restricted to the commercially available space, which is made available by Enamine. And this is one of the major advantages, right? That we are not going to complete custom compounds in the end that are often in the end generated by generative models, but we are in the end confining us to the commercially available space, which is more affordable and then also much faster available for delivery.
Great. Okay, I think there are some more comments. Okay, we're a little bit off time, but let's do the last question probably for today. So, how do you assess true chemical diversity amongst your experimentally validated hits from the virtual screen?
That's a good question. So, regarding chemical diversity amongst the experimentally validated hits, so when we do the screens and then in the end we get the ranking of the molecules, when we order compounds, we already look into chemical diversity, before we order. So we apply, clustering algorithms that, to make sure that, most of the compounds have different scaffolds in the end. And sometimes we also order, directly some analogs from which, from the library, like from the screen, because one of the nice features of the, the real space is that there are some, not too many, but some analogs contained. And this in the end can give us some initial SAR as well when we do the first round of experimental validation. And in the end, after the experimental validation, we have a pool usually of hits. And then we have in the end the choice, right, to choose how many scaffolds we want regarding further optimization and further screens and experimental validation. Yeah.
Okay. Thank you for the answer. I think that would be all for the Q&A session for today, because we're a little bit out of time. It was extremely productive, and I can't express my gratitude and my thank you so much for today's speakers, Selena, Christoph. Highly appreciate your commitment and your time. Thank you for joining us today, everyone, and we hope you stay tuned to our social media and not to miss the other wonderful events. And yeah, so stay safe, stay tuned, and we'll be in touch. Thank you very much once again. And with that, I'll be closing the webinar. Goodbye, everyone. Have a good one.